Okay, so I'm not a teacher and I have no idea what I'm doing, but I know that I do a lot better when I actually talk through things. So let's see if this is helpful. So this is just 7.1, but this is from the lectures and the books. So hopefully that's a little bit easier to kind of break it down, and hopefully you don't have to read the books if you haven't yet. So this is really all talking about enzyme inhibition and different qualities of that. So you have inhibition that can either be stimulatory or inhibitory. So it can either stimulate more production or inhibit production. So this is regulated by the bioavailability of the enzymes, which, if you follow this, is considered in RNA synthesis and processing. And when you think of processing, think of alternate joining of different sequences so that you have different protein products. And then also with protein synthesis, degradation, and targeting. You also have different things like control of catalytic efficiency that's also going to affect enzyme inhibition. So for control of catalytic efficiency, think about inhibition, allosteric control, covalent modification, and proteolytic processing, which over the course of time, it kind of gets more into every single one of those. So all of these are different forms of enzyme regulation, obviously through inhibition or stimulation. Cool. So for allosteric control, so this is kind of a part of what is going to be controlling your catalytic efficiency, you have metabolites, which are very small, that bind to regulatory sites on the enzyme outside of the active site. So here you have a little enzyme, you have your metabolite, it's bound outside of the active site, and he's going to be controlling shit. That's what the little graphic says. So in terms of inhibition, you have two different types. You have reversible and irreversible. So for your reversible, think of non-covalent. For your irreversible, think of covalent modification. So for your reversible, you're going to have it broken down into different types of reversible inhibition. So this is going to be quick restoration of the active site. So for reversible inhibition, it's used a lot in medicine because you're able to quickly have that active site exposed again. It's also good for things that are constantly shifting. So if you have too much of something and then you need to have some kind of control over that thing happen quickly, but then release, because when you have too much of it again, it needs to kind of go back and forth, if that makes sense. So you really want to think more about reversible control versus irreversible control, which effectively kills the enzyme, which we'll get into that in a second. So you can break this down into competitive, uncompetitive, and mixed. In the book, there's technically a third one that's non-competitive, but we don't really get into it too much in the lecture. So for your competitive, you're looking at this little graphic right here. So I can hold this up, sort of. Okay, so you have actual competition for the binding site. So you have the active site, which is going to be the primary site of this happening. For the other two, nothing is happening in the active site. Even in mixed inhibition, it's not active site. So you have your competitive inhibition, you have your active site, and you're basically, these two things are just competing for access to the active site. So then you have uncompetitive inhibition, and this again, we're not talking about the active site. But for uncompetitive, you have a whole different area in which this thing can attach itself. So you aren't actually having to compete for the actual active site. So this is outside of the active binding site, and it basically stops the active site and the enzyme in the active site from being able to make products. Then lastly, you have mixed inhibition. This is going to be the most rare of your inhibitions. It's also not active site, so it's other parts of your actual enzyme. And then you're looking at enzyme and substrate binding versus just substrate binding. So you can kind of look more at those little graphics, but I think the biggest thing to remember is just that you can break down your reversible inhibition into subcategories where you're either competing for the active site or not competing for the active site. If you're not competing for the active site, you might have something that's not competitive at all, and you can just have a little guy grab on there at a whole separate part of the enzyme. Or you can look at mixed inhibition, which is a mixture of things, but you're still not looking at the active site, and it kind of gets into more of that in the book. So then kind of moving over into some of the other stuff that we get into. So lastly, in the lectures, the last kind of thing that he goes through before we get into kind of what the book is adding on to this, is going to be your Km. So that constant is going to change or not change depending on the type of inhibition. So for competitive inhibition, you're going to have an increased Km, but you're not going to have a change in your Vm max. So you can see this down here. This is your competitive graph. So you can see that when you have low, normal, or high levels of your inhibitor, your actual Km is going to change, but your Vm max right here is not. It's going to stay constant. But basically in this situation, it's going to require different levels of substrate or inhibitor, because when you think of this type of reversible inhibition, you can actually overcome it by adding enough substrate. Because if you think about it, you have this competitive inhibitor, and so if you have so much substrate that your competitive inhibitor and your normal component that's going to attach that enzyme, you have enough substrate to allow for both, then you're basically able to overcome the effects of the inhibition. Cool. So then for your uncompetitive inhibition, you're looking at affecting the Km and the Vm max. You can see it in this Lineweaver-Burke plot. This is the inverse relationship, but you can see again that low, medium, high for the levels of substrate and the levels of the inhibitor. And then you also have your mixed, which we don't have a Lineweaver-Burke plot for really because it changes. It depends on the type of mixed inhibition that you're looking at. This one doesn't get covered super well. It's one of those things that like he made it seem like in mixed inhibition it's going to bind to the active site, but then in the book it's very clear that you're still not looking at active site inhibition. Okay, so things from the book so that you don't have to read the book. Cool. So reversible inhibitors can be decreased by diluting enzyme reaction versus irreversible, which is going to be unaffected. So again, with reversible inhibitors, there's a way to reverse it. For irreversible, there's no way to reverse it. Kind of a way to think about it. The example used in both the lectures and the book is for malonate, but they don't really put that in perspective. So malonate is the reverse competitive inhibitor of succinate dehydrogenase, but this is actually a really important component of the body, but doesn't really go into it. So succinate dehydrogenase is basically involved in catalyzing oxygen. So it's able to be part of the Krebs cycle, and it actually is able to catabolize oxygen and then add that into your electron transport chain. That is super, super important for that final step of generating ATP. So that's like where you have that electron transport chain and it ends in a little oxygen, and it's just like collecting oxygen substrate stuff through oxidative phosphorylation. That's kind of like the component of it, and then it makes ATP. Not an expert on that, but essentially it's a really important component. So if you're inhibiting it, you're inhibiting essentially the production of ATP by the end of it. So it is an important thing to think about when you think of why are we talking about malonate and succinate dehydrogenase. It's because succinate dehydrogenase is involved in the production of fumarate, which is involved in the Krebs cycle, which is involved in the electron transport chain, or the electron transport chain is a part of, which is going to help us produce ATP. So that's why they use that one, and that's a reversible competitive inhibitor. Cool. So that's basically what this says here. Keep picking it up. All right, so then kind of moving on to the last little bit of this. I feel like this took a long time to write, not a long time to talk about. Oh, move over here. Okie dokie. It's a very big whiteboard. Okay, so last really thing is that the irreversible enzymes effectively kill your enzyme. So it uses chymotrypsin, again, to talk about things, but if you remember, your catalytic triad includes serine. So the disopropylfluorophosphate is the example they give you for your irreversible inhibitors. And what it does is it basically is able, with anything with a catalytic serine residue, inhibit it, but in an irreversible way. So it binds super, super tightly and covalently, which is the important difference between irreversible and irreversible. So it's a covalent bond, and it effectively makes that whole chymotrypsin unable to do what it is supposed to do, which is kind of goes through that whole mechanism that we talked about, but it starts with that catalytic triad. So then for the inhibitors, just to go over a little bit extra stuff that the book gave us, for competitive, you bind directly to the active site. You can obstruct the active site, or you can basically bind elsewhere that causes a conformational change in the active site. So there actually are more ways than just binding directly into the active site and blocking the whole thing. So it's just something to think about, but when you see active site, you should definitely be thinking of competitive inhibition. So then another thing that it adds is thinking about how tight is the inhibitor bound to the substrate. That's a really important component, or bound into the enzyme active site, into figuring out, is it effective? And so the equilibrium dissociation constant between the enzyme and the inhibitor, so this is kind of like your dissociation constant, your KD, but it's Ki, because it's specifically the dissociation between the enzyme and the inhibitor, versus with KD, obviously it's a little bit different. And then your competitive inhibitor effectiveness depends on the concentration and the affinity. So it introduces Ki just to think that you're thinking both of the concentration, just as we said, if you have a lot of substrate, you can overcome competitive inhibition, but you're also thinking of your affinity, how tightly do they bind together.